Hello, my name is Justin, and today I'm going to be presenting to you my data analysis done on an experiment to do with microbe diversity within personal objects. So, first off, what is this experiment that we're doing? This experiment is we're looking at to answer two questions. One, do individual differences lead to different microbes being present within humans? In other words, do the differences between different humans mean that they have a different makeup of microbes? And secondly, we're looking at if different personal items we come in contact with from our day-to-day also have different microbes on them. So things like your water bottle, shoes, wallet, along those lines. If there are different microbes, that would indicate that different parts of our bodies possess different microbes. So why are we doing this? Microbes are everywhere. They're all over our body, inside us. And for a long time within human history, we were not aware that they existed. But not only were we unaware of their existence, we were also unaware of how much they help us. A lot of people see bacteria and other microbes as dirty or being unclean and leading to disease. And that may be true in some instances, but we couldn't survive without microbes. They regulate our bodies and protect us from disease. They make the earth habitable by driving nutrient cycles, so ensuring that we always have enough nutrients. And so, as you can see, they are an incredibly useful organism, or type of organism, which we need. And so, understanding them better and the types of microbes that exist in different places can be very, very useful in further understanding ourselves, as well as just leading to a better world, potentially, by harnessing what the microbes are doing within medicine. So, what is the experiment? So, we've taken microbes from 5 different people and from up to 5 different items that those people had. So, inside of shoes, water bottle mouth, phone screen, reading glasses, and wallet were the 5 items we were looking at. The gender breakdown, we had 3 males and 2 females within our study. Ethnicity, we had 1 European, 2 Australian, 1 Southeast Asian, and 1 Middle Eastern. And we had a negative control. How was it performed? So the cotton bud, which we used to get the sample, was sterilized firstly with ethanol. We then swabbed each item with the sterilized cotton bud, and they were stored in a test tube, and certain processes were done on it, as you can see on the slides, to ensure that it was a clean sample. And then we used a technique known as to extract the DNA of each sample. So in other words, what I was doing was it was reading, looking at the sample, looking at the DNA, and then creating a list of the nucleotides. So we take up 4 letters, GACT, which make up everything, including those bacteria. So by analyzing that, we should be able to understand the structure of each bacteria or microbe. So were there any issues with the DNA extraction? This is a very important question to ask. First off, no traces of DNA were found on the negative control. Cotton bud, which was the cotton bud that was dipped in ethanol and dipped in nothing else. This is great. This means that our negative control worked. So that's good there. So no issues on that front. However, 5 of the 23 items failed to produce a sample for unknown reasons. We're not exactly sure what happened there, but that should be noted. However, everything else was fine. The 18 other items were perfectly okay. So let's get on to the data analysis. Now, first off, what we're looking at here is the quality of the DNA sequences we've got. With the Illumina method, it is fantastic for reading DNA, but it is not always perfect. Things can go wrong, which can lead to some errors. And what we're looking at here, we've got 2 graphs, one looking at the forward read of the DNA, that is the top graph, and another one looking at the reversal reads of the DNA. That is the bottom graph. That is because DNA can be read both forwards and backwards. Now, if we take a look, firstly at the forward read. Well, first off, let me explain what the graph is. The y-axis here, that is what is known as the Phred score. Spelled with a PH, not an F. And what it means is, so from 10 and above, everything 10 and above is 90% good quality, so which is a fairly high score for most people. However, since we're biologists, we don't just want 90%. So if we look at the 20 and above, there we're dealing with 99%, 30, 99.9%, and so on. So with this, we want to take a score so that we're getting the most accurate samples possible. So we looked at the forward reads, and we saw that about 240, everything's above. Most of the things are above 200. Look, almost everything is above 20 there. So we took 240 because we want it to be 99% accurate, and then we did the same for the reverse reads. So we went to 20, looked for the dark place, which indicates a lot, and found that about 200, most of them have a Phred score of 20 or above. So those are the two scores we took. So we should have data that is 99.9% accurate. Now, of course, this is a balance. We are losing some data, unfortunately. However, the data we have gotten is hopefully of top quality. So there is the trade-off there. So let's take a look at the data left over. So you can see there's a minimum frequency of 0. This would be our control, a negative control that would have gotten this. We've got a good median of 390 and a mean of 366, so indicating there's some level of outliers which throwing the mean off. So some are quite large, others are quite small. So it is useful to use the median then for any further analysis we might do. Next step, looking at what actually we have. So we can look at this graph, and we're able to see that the pink parts of the graph are bacteria, and the green parts are unassigned DNA that we're not quite sure what it is. So most of the samples are in fact mostly made up of bacteria, which is fantastic. That is exactly what we want. It is important to note that this one here, AP21, is quite high, as well as AP8 and AP4. They are higher than the rest. However, it shouldn't make too much of a difference. As well as, if we look here, we've got our two controls here with nothing. So that, again, is what we want. So yeah, let's get into the further analysis then. Next, we're looking at p-sampling depth. What we want here, similarly to before, where we looked at looked at refracting, not refracting rather, where we cut off some of the data. We're only a high quality one. Here we're looking at the sampling depth, and we want to find the best number. So right here, the sampling depth, so that we're able to take a decent number of samples here, but also we don't want to lose too many different sequences seen here. So as we can see, the blue parts of the graph indicate that this is kept and this will be used in the future analysis, whereas the grayed out white parts indicate that it's not. So of course the negative controls, because they have no data, any number above 0 would rule them out. Now we have lost a little bit here on the water bottle, but as I said before, that is part of the trade-off. We lost 15% of different samples. However, we were able to retain 33% of the data, so we've got a good sampling depth. So I'm quite happy with the trade-off there. Let's move on to the next thing, which is alpha diversity. What alpha diversity is it measures the abundance of different microbes within our samples. So what we're interested in is A, do we have enough samples, and B, for any other analysis to be worthwhile, and B, is there a difference in the abundancy between different things? So let's take a look. First off, we have our alpha refraction. It tells us if we have enough samples. Now what we're looking at here with this graph is we want to find the bit where it levels off. Leveling off indicates that we have a good amount of samples, and that we do have enough samples for us to perform any analysis. So you can see here at a bit over 200 is roughly where it levels off. So that's good. It definitely means we've done the right thing. We have enough data. So let's keep going then. Okay, next what we're looking at here is we're looking at the difference between DNA, so the abundance, on the items. So the 5 items mentioned, the glasses, phone, shoes, wallet, and water bottle. Now what immediately stands out on this graph is that the wallet is much smaller, so smaller standard deviation, but also it is far away from everything else. It appears to be significantly different. And when we check the figures, the maths behind it, we do find that there is a significant difference in the abundance of the wallet in comparison to the rest of our items. The rest of the items all are of a similar level, even though we can see slight differences in the graph. There is no significant difference between them. But the wallet is significantly different from them, and there appears to be a greater abundance of microbes in the wallet, on the different wallets that we looked at compared to the others. This is rather interesting. So again, at the moment, all we can say is there is more on the wallet than anything else. We can't exactly say why, although we could make a few conjectures, but they would not be backed up in evidence. Well, let's continue looking at the different amounts. So we've got people now. So the difference in abundance between people. So these are all pretty similar. We do have rather high p-values, and you can see on this box plot here that they're all relatively close together. So yeah, there's no significant difference here. It doesn't matter based on people. It also, once again, no significant difference based on ethnicity. And as you can see, sex is incredibly close together. So what we saw there was that in terms of abundance, there was no significant difference when it came to individual differences. So sex, ethnicity, and person. They all had around the same amount of alpha diversity, or the abundance of microbes. However, when we looked at items, we did see a difference between wallet and the rest of the items, indicating that there is something going on there where the wallet has more, a greater amount of microbes than the rest of the items. Now that we've looked at abundance, let's look at difference. So is the DNA of these different the samples we've looked at, is there a difference, or are they all just the same type of stuff? Let's have a look. So this is what we call beta diversity. So what we're looking at is difference on the phylogenetic tree. What that means is when it comes to classifying different species of different things, we look at the phylogenetic tree. In other words, how varied they are based off different features. So we're combining that with our DNA to look at beta diversity and see how different the DNA we've sampled is from. First off, what we have here is the emperor plot. It should be noted, more emperor plots were created, detailing other differences. However, it wasn't found that they were helpful. And so, in the interest of time, I've only included this one. Now, what this is showing us here is a principal components. So we've got two components, actors 1, which explains 12.71% of the data, and axis 2, which explains 10.76% of the data. Sorry, of the variance. So the axis 1 is slightly more important, so we should be looking more at the x-axis than the y-axis, although the y-axis still has some level of importance. Now, what we're interested here in is seeing if, I've got a color-coded items here on this legend, we're looking to see if any clusters form, if any items, when they're placed together, form a cluster. On the other emperor plots that were created prior to this presentation, it didn't appear that there were any clusters when it came to individual differences. However, if we do look at the emperor plot here, when it came to, when we're looking here at shoes, you can see 1, 2, 3, 4, 5, that the 5 samples of the shoes are all quite close together. So there does appear to be some clustering. This indicates that there probably is some sort of difference between the shoes and the rest of the data. So it is worth further investigation. So then let's have a look at our pairwise per maneuver results. As you can see on the second column from the right, we have our p-values. We've decided that if they're less than 0.05, then we'll reject the null hypothesis. Now, there does appear to be an error here where 0.093 has been circled. However, it should be 0.026 that is circled. The PowerPoint itself was working, but during this recording, for whatever, and the recordings before that, for whatever reason, the 0.026 was not circled. Anyway, so we do have 3 instances of significant difference, so less than 0.05, and all of them occur within the shoes. So it does appear that the inside of the shoes do have some level of difference between the rest of the data in terms of the types of microbes that are living there. As well as these pairwise per maneuver results were created for the other differences which also grouped together as under the term individual differences, and none of those were significant. The only significance we saw was the difference between shoes and the other items. So once again, very interesting. And one of the limitations of this is we're able to say that there is a difference between the shoes and the rest. However, we're not actually able to say where that difference may be. So in terms of future directions, we might want to look at more depth in the microbes found on the different items, in particular wallets as well as shoes. Wallets to examine why there's so much in comparison to the rest of the microbe sites, and shoes as there might be. We want to know what makes it different. Why are they so different from the rest? So looking at the types of bacteria that exist on shoes in comparison to other items would be very useful in understanding this better. Also, it could be worth repeating this again, looking at other personal items and further hone down. For example, if we were to look at socks, for example, which would be closely related to shoes, we'd want to see if there's a difference there. Or maybe there's not, and that would also be interesting. As well as other, because clothes do cover most of the body, they could show us many different things, further things that we got from this experiment. Finally, it is important to confront any potential confounding variables in this experiment. Now, it was done. Now, mostly it is fine. It is was conducted in quite a scientific manner. However, in terms of the subjects we had, all of the females within the study had the exact same ethnicity. They were all Australian. So there could be that could potentially be an issue. However, based on what our results have shown, it doesn't appear that there is, in terms of the microbes, there is a large amount of individual difference based on this experiment. So, while yes, that could be a confounding factor, I would say it's not there is a low chance of there being an interaction effect where ethnicity and gender interact to create some type of effect. However, based on our data, that does appear unlikely, but we can't say for certain. So that is my data analysis based on this experiment. Thank you very much for listening, and have a great day.